He did his PhD in neuroscience at the International School of Advanced Studies, and he did his postdoc just down the road at UBC. He's been at the University of Lethbridge since 2013 as a faculty member, and his lab studies the physiological and computational basis of sensory perception and learning using in vivo imaging methods. Today, he will present on hippocampal-cortical interaction, memory replay, and default mode network. So thank you so much for being here for one of our first INN seminars, and looking forward to your talk.
Thanks, Randy, for the invitation, and Brianne for hosting me. Let me talk a little bit about the work that I've been doing for the past several years or so. So it would be a bit of a jump from one field to another. So I wanted to kind of give you a heads up. So it may be a bit of jittery, but I really also appreciate if you kind of interrupt me wherever you think I didn't convey the message or it was not clear. I really like to have this kind of interactive meeting. This is not working. Oh, good. And I have to use perhaps my pointer for the people that are in Zoom audience. These are the people that kind of work in the lab and contributed the work that we have been doing for the past several years. These are the funding, and these are the people that I collaborate with, in particular. Some of the individuals are Ian Bishop, Bruce McNaughton, Rob Sutherland, another fellow who is not in the picture, Brian Cole. I do really work, but I've learned so much from them for the past several years. So this is more of a contribution to their knowledge. So I guess I may not necessarily need to explain to this audience that brain use multiple memory, long-term memory system to store memory. Declarative and non-declarative. If the memory is kind of rapid, unconscious is more non-declarative memory, and there are different regions of the brain that are involved. And there are some declarative that is rapid and fast and requires some accommodation of facts. So in the case of HM, as you may know, because of the damage to hippocampus and surrounding area, he got some problem with declarative memory, in particular episodic memory. And what is really episodic memory? This is something that I really like to emphasize early on at my talk. Episodic memory has 3 different features: where, when, and what. So I don't know why this axis is not coming. Well, yes, when, what, and where, and if something that has to be related to me. Okay, just give you an example. I am in my birthday party, and who comes first? Who comes after? What's the background music? What's the type of food there? And what happened when those 2 friends got into argument? That's a kind of episode, right? And that episode have this trajectory in 2 dimensions: when and where and what. And that has to be related to me. It's personal. There would be another episodes that also has that similar content, birthday, but at different time and space, and different space. And then several of those episodes have similar context, related to myself, such as a birthday, that becomes a semantic memory. So that's the difference between semantic and episodic memory. If you strip off the content, where, when, what of episode, that becomes semantic and become abstract. So it's common to everyone else. But episode is really things that is related to me. And that's what we know. It relies on medial temporal lobes, hippocampus, and surrounding areas. And We all know that in order to form the memory, usually sensation and information comes through primary sensory areas, then travel up to more higher association cortices, and from there it goes to the hippocampus to create an episode or make a map there. So you can basically make it upside down, make it like a pyramid. So at the base of pyramid, you have a sensory cortical areas, and then you have association cortex, and you have hippocampus. The basic elements of those circuitry are the same. They have this recurrent network that could basically give them a possibility to record or store memory. However, and that's basically why if you lose hippocampus, some of the information get lost because these other areas are not able to communicate with one another more efficiently. So they become isolated pieces and they cannot be pieced together. I really like to talk about theories, and theories that are basically built up when there was no experimental evidence. And when it comes to hippocampus and memory, I really like this particular hypothesis of theoretical paper came along in 1986 by Taylor and Desena, famous for hippocampus indexing theory. So what they argue is that during the this is the neocortex, this is hippocampus. During the formation of memory, certain moduli in the neocortex get activated at different area that create an index within the hippocampus, which can be reactivated during sleep and otherwise. That's when the memory get formed, memory transform. However, during a memory retrieval, if you provide a piece of that information, just a piece, not all the episode, the background of music or taste of coffee or something, that could perhaps create or activate the index that was already saved in the hippocampus, and activation of that could recreate the entire index, which in fact can be sent back to neocortex, in which neocortex used to recreate that episode, right? So that's what we know about, that's what is being proposed in hippocampal indexing theory, and that's in 96 with not much of information and theoretical understanding or experimental tools. So this part is kind of very well known to pattern completion. So you have a piece of pattern, partial cue. With that partial cue, you can basically reactivate, recreate the entire episode, and that's often believed to be happening through this hippocampal neocortical interaction. This is what I really like about the memory system. So in a way, hippocampus is like a pointer. Doesn't really store the memory, perhaps a little bit of that. The information is stored in the neocortex. Hippocampus become like a pointer, like a librarian. Doesn't necessarily know and have the knowledge of the word, but knows where to find that knowledge. So it can point it out and it can basically link to each other. So that's basically what it comes to episodic memory and the role of hippocampus. So there are some prediction from indexing theory and what it makes a good index code. To have a good episodic memory, the index code need to be unique to each stored memory with minimal interference. You don't want to have memory mixed up, right? That's a basic element of that. Now there's a good experimental data found in hippocampus that hippocampus, such a circuitry, can really generate a unique code compared to any other region in the neocortex. The index code also needed to be retrievable by auto-association or pattern completion. I talked about that. For that, the code need to be sparse. And again, that's something that hippocampus is very famous for. Is the most sparse. It could generate a sparse code, which actually changes when you go from different hippocampus to a different part of hippocampus. It also predicted that the code need to be widely distributed in the neocortex, because there are different sensory areas, there are different pieces of memory that has been stored in different area. So it has to be broadcasted globally to the neocortex. But there's a biological constraint. We know that hippocampus has only certain biological connectivity. And this has been a puzzle until not that long ago. And this is something I'm going to talk a little bit about in my data. And of course, also it was postulated that to consolidate the memory, the index code needed to be spontaneously reactivated while the brain is in offline mode. Okay? This should cause reactivation in the neocortex. Reactivation of memory in the hippocampus should cause reactivation in the neocortex, and that is important for consolidation of the memory. And last, the index code needs to be stored within cortical module along with the episode attribute, because index is just index, but then the content, the attribute, it's stored somewhere within the cortex. So this should be basically somehow a coordination between index and context. And this is something that I've been very much interested for the past few years, arguing that perhaps index is stored within superficial cortical layer, and attribute is stored within deep cortical layers. And there has to be some communication between this columnar circuitry in the neocortex that could recreate the entire memory. It's a work in progress, and I don't think I will be able to show some of the data. I mean, it's preliminary data, but I'm happy to discuss about that later on. So the indexing theory was proposed in 1986, as I said, but it doesn't really provide or make any attempt to link what was known about neurophysiological behavior of hippocampus, which was very well known by early 70s by the work of Vishal and Van der Wolf and many others that came after. So what it is that there's a very nice dichotomy in hippocampus between electrophysiological characters of brain oscillation and animal behavior. So when the animal is walking or is in a particular engagement in a task or during REM, the hippocampus generates this nice oscillation called theta, right? When the animal disengages from the task, involving consummatory behavior such as eating, or during non-REM phase of sleep, the hippocampus generates this irregular type activity, and within those time, you can see sometimes this very big burst of activation. It's called shuffle ripples, okay? Or in summary, we can call it ripples. So ripple is the most and strongest oscillation that is being generated in the brain in terms of the number of neurons that are involved and also the strength of the synchronization. This is an example of that. So this is ripples, only 50 to 100 milliseconds, and now we know that it's involved in memory consolidation. People have done so many works on that. However, Yuri Buzaki came along in 1989 and came with his two-stage model of memory transformation, in which argued that during encoding of memory or experience, hippocampus shows theta activity and hippocampus and neocortex interact with one another to form the trace of memory trace. And during sleep or when the animal disengaged from the task, hippocampus transition to this irregular activity which generates ripples, which are elements of, which is a kind of work as a courier signal, a memory courier signal, indexing courier signal. There's a time that information passed along to the neocortex, and that caused reactivation of cortex and hippocampus at the same time. And so this is something that was hypothesized, and soon after, many experimental evidence came along, in particular, looking at rodents navigating from one place to another. As you may know, if you recall from hippocampus and let the animal run from one space to another, there are cells that are getting activated at different locations. There are these cells that are activated at this part of the maze, these cells that are activated a little bit later, and so on and on. So when the animal goes there during disengagement of the task or during sleep, when shuffle ripple triggered in the hippocampus, we can see the same code that was happened during animal behavior can now reproduce, but in a very compressed format. Okay, so this is the action of hippocampus, there's a replay, and this has been shown that we can see replay and pre-play in cortex and hippocampus, and now we know that they have to be coordinated for memory to be consolidated. Well, I mean, that's all fine, but still the major prediction of indexing theory was still unknown due to technical difficulty of recording cortical activity. And so this is something that we began to work on early on when I moved to University of Lethbridge. And the idea is hippocampus cannot, because of the biological restraint, anatomical input that goes from hippocampus just activate piece of the neocortex. And by itself, also cortex has some limitation because each module is loosely connected to the other module. I mean, we think that cortex is all very well connected, but if you just find two neurons in the neocortex, the chance of them being connected is 1 to 1 million. So it's very sparse connectivity. Again, that brings really a major theoretical restraint in how the brain can, encode information that is coming from the hippocampus across the cortex. So Francesco Battaglia, I think he used rat and tried to map the hippocampal output using electrophysiological recording. Uses a lot of electrode and in fact created a lot of hole in the brain. That didn't go very well. They couldn't really get much of information. However, this paper came along in 2012 by a group of logothesis in Germany, which was quite novel and surprised by a lot of people that been working on hippocampal-cortical interaction for a long time, which was mainly limited to rodents, or at least in part. So what they did, they had a macaque, anesthetized macaque. They had an electrode in hippocampus, and what they did was to record shuffle ripples and then try to see what the pulse signal looked like before and after shuffle ripples. And that's a summary of the findings. So they argue that when there is ripples in the hippocampus, most of the neocortical area get activated. And they find this nice dichotomy. So most of the subcortical area get deactivated, and most of the cortical area get activated. Well, this was a big surprise, but also created a lot of reverberation within the community. A lot of people say that's not true, because we know that if you record from subcortical region, they're not deactivated, they're actually active. So that was again the main drive of us repeating those experiments this time around with perhaps techniques that provide a bit better temporal spatial resolution. And that's what we do in the lab. We use imaging, optical imaging, and the advantage of optical imaging when it gets combined with genetic is that you will be able to image many different neurotransmitters. We can look at inputs to each area. We can look at GABAergic input. You can look at glutamatergic input as exciter drive or GABAergic as inhibitor drive. You can also look at all the neurotransmitter and neuromodulator that I will show you a bit later on. At the same time, so you can look at the neurodynamics by measuring the memory potential or voltage. And at the same time, you can also record the output of individual neurons. So you have a kind of system that you can look at input, you can look at output, and you can try to infer what's happening in the neocortex. Unfortunately, though, we cannot at the moment image many of them at all at once. So it's one preparation, we image them differently. And I think within the next 2-3 years, we will be able to do a bit of multiplexing, being able to image some of those things together, like GABA and glutamate and calcium, and perhaps one of those neuromodulators like a norepinephrine or dopamine. So this is my methodology slide. We use mouse as an animal model. We create a window like this one over the cortex. We use some kind of electrode, different format to record electrophysiological signals, kind of spikes, mainly from hippocampus. That's where we detect sharper ripples or ripples from different layers. Then you can either record brain activity in wide field mesoscale imaging, which provide a population imaging of many neurons from big area of the neocortex. This is almost, half of the neocortex. This is bregma, this is midline area, this is visual area, this is auditory area, this is frontal area. So you can see all of that advanced with a great temporal spatial resolution. Or what you can do, you can zoom in a small region of the cortex, and in fact record individual neurons. So this is 2-photon imaging of neurons within a retrospinal cortex. You can see this little guys blobbing, showing light. So these are calcium flux that get activated when those neurons are firing. Okay, so now you can say I can move in different scale, in time and space, to be able to understand what's happening when hippocampus, for example, is generating index or shuffle ripples. The animals are we record the animals at different behavior state, either when they are asleep or awake, which is the biggest transition in the brain state, or when they are engaging in different behavior. So some of the behavior that we have been using are related to navigation, and it's a little bit funny because we had fixed the animal because of the imaging. So what we have a treadmill that the animal can walk voluntarily whenever they want, and they go from environment. They basically explore the environment. The environment is either visually driven or is tactile driven. The visual driven animal are basically can walk on the environment. It's a VR system. So the animal goes from one room to another, and the idea is to be able to record, for example, from hippocampus to understand where the place cells is. Or you can basically have only tactile information, again, different sensory stimulation. And the idea is to, for the animals to understand where the tactile cues are, and based on those tactile cues, they can understand where they are, in what stage of lapse they are, and where they can start basically getting a reward. So this is what we use. And to make the story short, we start Jawad carrying a PhD student in my lab, did simple experiment. We got it from hippocampus and simultaneously image the neocortex. This is a movie of his recording. This is hippocampal LFT. This is when he filtered it out, so you can look at the shuffle, and this is brain activity. Simply, what he did, he isolated the shuffle ripples, and then he lined them up and made a map of neocortical activity during shuffle ripples, exactly what logothesis is, and surprisingly, so a little bit different than what they found there. So this is a movie. Let me try this a little bit slowly, so you can see that this is center shuffle ripples. 0 is a shuffle ripples, and this is 1 second before and after that. So what you see, there's different level of activity. This is the average of many shuffle ripples, maybe 600, 800. So there's different area of the brain get activated or deactivated. So before shuffle ripple, this area of the neocortex get deactivated, and then when we go to the center of shuffle ripples, you see this activation. So let me also explain that the warmer color means more depolarization, and the colder color means less depolarization. So this is glutamate means, sorry, this means more glutamate or less glutamate. You can do also voltage, you can do calcium, which is the result are very much similar. So at close to the center of shuffle ripples, you see that some region of the neocortex are getting activated. So there is a midline area, retrospinal cortex, and there's this temporal area that are getting activated. And then you can see this evolve, and then and then it goes back to the normal and the activation. So unlike those simple activation, deactivation that was proposed by Bohr, it seems that the signal are much more dynamic. So there's different region of the neocortex getting activated, deactivated, different temporal dynamics and gradient. So what Jawad did was to average them up. I mean, he did many different types of imaging, in sleep, either glutamate region, voltage, calcium. To make the story short, I'll just focus in the summary. So this is summary of all the recording that we did, and we could show that there's a gradient of cortical activation. So certain regions never get activated, and some regions get highly activated, or activated early, and some regions get activated later. So this is a map of area that is recording, parcelized to different regions. And you can see again, these areas are the ones that are getting highly activated. And I just wanted to bring up this figures taken from a paper published recently in Cell, showing default mode network in mouse. So as you can see, almost the same area that we see activated during shutter ripples are showed up in the mouse default mode network. So I wanted to argue that it's safe to say that these two maps are almost identical. So you can call it default mode network, or you can call it memory mode network. That's something I really like to call. So well, I want to skip this, but there's sharper ripples, but there's many other things that is happening in the campus. There's gamma oscillation, there's lower oscillation coming from different areas of the neocortex. So with this technique, you can map them up. There's the data is already in this bioarchive paper. So if you're interested, you can go up and look at that. So big part of my lab does Alzheimer's disease, or use mouse model Alzheimer's disease. So we have, I think, 5, 6 mouse model. Majid Singh, a PhD student in the lab, using exactly what Jawad did, tried to record the sharper ripple trigger cortical activity in control mouse, or the mouse that has some genetic mutation to show behavior deficit. So we have published the behavior deficit, but what I want to bring your attention to is that what we found was certainly there is a change in a pattern of sharper ripples in this animal before they even show the behavior deficit. But more importantly is that the way that the cortex gets activated at the time of sharper ripples. So this is a control animal, and this is the animal that has been showing has got some genes that taken from human, and you can see that the hippocampal-cortical interaction is being totally impaired much earlier than the animal shows any kind of behavior deficit. So I think we have looked at Alzheimer's disease in many different concepts. Even the animal does exercise, cognitive training, stimulation, and stress, and we use more the similar kind of imaging to understand how each one of those either accelerate or decelerate the onset or strength of impairment in muscular Alzheimer's disease. So I will just pass on. So what Jawad did next was, instead of looking at single sharper ripples, he basically looked at the bundle ripples. So just think about analogy. So when the mouse is going from one space to another space, if the path is complicated, instead of having one sharper ripples, you need several sharper reservoirs, several elements of sharper ripples or index that is going to be broadcasted to the cortex. And in fact, we know that when the memory gets reactivated, you usually have several of those sharper coming together. So what we saw that, yes, one single sharper ripple is getting, kind of smaller area of the neocortex. When you have more than that, you get much larger area of the neocortex. And so this is something that you're writing a paper arguing that hippocampus is really sending information to the default mode network, we call it memory network, and that's where the information is broadcasted to most of the other area within the cortex, but also within the brain. There's some evidence that we have, and I'm happy to share later on. And this is something that we're hoping to also get into stimulation and modeling. Anyhow, so Jawad, instead of just looking at average signal, he kind of looked at the sequence of cortical activation. So what he did, he sorted all the sharper ripples. So I don't know, there's 1,800 in single animal. And this was exactly where we find a bit of surprise. So the dogma was, hippocampus always lead hippocampus, always. But what he found was that most of the shuffle ripples are actually preceded by cortical activation, and only a small subpopulation of those are following the shuffle ripples. We have a bit of theory to explain that, which is purely speculative. So what we argue is that for recent memories, or memories that are formed, which is not that much trivial if you're taking the animal just to image them, it's just a normal ordinary life. So for the recent memory, what happens is that the neocortex get activated, or activity within the neocortex create the sharper ripples, create the index within the hippocampus, which represent itself as a sharper ripples, and those sharper ripples backward-propagate to the neocortex, which get cause activation in neocortex. This explains this part of the spectrum. However, for very remote memories, the memories that are formed long time ago, most of the things is actually consolidated within the neocortex. So activation of neocortex causes this K complex, which we know it's happened during sleep. And when it gets activated, it creates the index within the hippocampus, which represents this. And this is again purely speculative. And Jawad and Milad are basically trying to design an experiment to test this. Basically, what they do, they have a mouse, they record the brain cortical and hippocampal activity before formation of field memory, and they bring the animals and recreate that experience and let the memory get older and older, and then they add new memories. So the idea is to see whether a bit of temporal dynamic within neocortical and hippocampal activity. Again, I'm not going to go into the detail. This is already a big paper, but just wanted to show you that just a day before delay-free conditioning, as clearly you can see that neocortex is activated before the center of shuffle ripples. However, the day after learning a new episode, you see that hippocampus clearly preceding the cortical activity. But again, there's a bit more onto that. I mean, how different area of the neocortex getting activated for certain memories are something that they are working on, combining wild field imaging, but also looking at neurons using 2-photon imaging. Perhaps I have to kind of skip this, again, too much detail, but just wanted to show you that sharper ripples, when get activated during awake, is very different than when the animal is during sleep. If you remember, during sleep, the default mode network getting activated. However, when the animal is awake, the network get deactivated. And again, we were able to image different layers of the neocortex, and we know now that this deactivation happened earlier in layer 5 than layer 3. And this is again goes back to this hypothesis that we have in which the attribute is stored within layer 5, while the index is being stored in layer 3. And if they come almost at the same time, there's going to be interference of memory recall. So I wouldn't go into the details. What I prepared this only for because I knew Randy likes this, so I just add some of these slides to kind of show some of the work that we have been doing for the past 3 years or so. I'm really interested, and this has been going on for a long time, to understand the role of neuromodulators. I mean, and everyone talk about dopamine, serotonin, norepinephrine, and acetylcholine and histamine. There are many of those. It's been very difficult to study them because they are coming from small discrete of neurons. Their connectivity is not necessarily very well known except certain regions. And the release is also very complicated. And the only tools that has been used was, pharmacology mainly to study them. However, recently with the revolution that came along in genetic, you can genetically isolate individual neurons that release different neurotransmitter and insert different sensors within those individual neurons and use that to image them while the animal is involved in a task, or also try to understand their connectivity or just basically their structure, depending on the sensor that you insert. These are the animals that we are using, crossing with different animals. It's expensive for sure, but it's worth really understanding what it can give us. And we use also a bit of machine, fancy machine, in which when you insert those sensor, you can look at individual point at the level of nanometer to understand their connectivity. So just give you an example, what we did, we have looked at cholinergic connectivity across whole brain. I'm just showing you how it looks like within the neocortex. Okay, so if you look at the cholinergic connectivity within neocortex, the area that we image using functional imaging, you see that there are lots of connection of fibers from basal forebrain into the midline area, but there's less activity within this sensory area, a little bit less in the auditory cortex. Again, this was not necessarily known until now, and if you basically adds up, again, kind of show that there are some part of the default mode network that is showing up here. If you're doing this exactly for norepinephrine, serotonin, and dopamine, but in addition of that, we are using the same methodology to map up this misfolding protein in Alzheimer's disease, like where is the tau, how it's being progressive in different region of the brain, or how amyloid basically spread within different area of the brain, and how different factors can modulate those. This is a machine that generates a lot of data and something that not to make us busy for a long time. However, I mean, structure is a structure. It's just very, you don't see any dynamic, and I like dynamic. So what we do, we like to look at dynamic of acetylcholine within the neocortex. So here, what you're doing, you're imaging release of acetylcholine within the neocortex by the means of expressing calcium sensor into cholinergic neurons. So these are axons of cholinergic fibers coming from basal forebrain. This is the cortex, and this is the hippocampal recording. This is the showing what stage of state the hippocampus is, and as you can see, the dynamic is a little bit different than what often people propose. Neuromodulator used to be considered a scalar quantity, meaning is on or off, more or less like that. But this this data suggests that it's not really a scalar value; it's a vector, which has a different trajectory in space and time. And again, this would complicate a lot of people that are involved with simulation of brain activity like Randy. I think it's going to make them, and this is the same thing, but it's not the same. If you look at neuropinephrine, it's totally different things. This is a hippocampal recording. So what we do here, this is in a sleep. This is in a sleep. So what we do, we divide the frequency of 5 Hz to total frequency range. So just isolating when the animal is in REM or non-REM. So here, if the ratio is low, it's in non-REM. As soon as the ratio goes higher, it becomes REM. So you can see how acetylcholine level changes in the neocortex when the animal moves from one state to another state. Yes. I mean, you in REM that there's a high level of acetylcholine, but the switch is much more dynamic than what is being taught. And again, you can basically do this, you can measure those things, and you can add them up, the acetylcholine levels, and you can do it in many different animals. And again, what you see is this area that we know getting activated as we know as default mode network. So again, concentrated within that area. Okay. This is acetylcholine, just acetylcholine. But just to make things complicated, we image shuffle ripples and look at acetylcholine levels. Okay, so this is acetylcholine just before shuffle ripples. This is center of shuffle ripples, and you see that at the time of shuffle ripples, there's a clear antiphase activity between acetylcholine and shuffle ripples. And now, if you look at norepinephrine, it's totally opposite things. So you see that at the center of shuffle ripples, there's this in-phase activity between ripples and norepinephrine. And again, although activation of cholinergic and norepinephrine both involved in default mode network, like here and here, but when it comes to the shuffle ripples and their temporal dynamics, they're totally out of phase. Again, this brings up, perhaps acetylcholine is for segregation and norepinephrine is for integration of memory storage or memory recall. But again, these are the things that are hypothetical, and there hasn't been really much of experimental evidence to bring those up. So, until now, we use wide field imaging just to map out what area of the cortex get activated with the shuffle ripples, but we didn't really have a cellular resolution imaging to understand where the index is and how it's being broadcasted to the neocortex. To do that, we did two-photon imaging, so you can look at individual neurons when the animals actually get involved in the behavior. So the story short, this is a mouse running on a treadmill. It's a different system. Sam is a postdoc in the lab who's really interested to discover these conjunctive cells that encode either space or time or context or content. So that's what he's doing. I'm just bringing an example. If you recall from hippocampus, while the animals navigate through the treadmill, you can see cells that are activated at different space, a different location. This cell is activated at the beginning of the treadmill, and this is cell that is activated at the center of the treadmill, and this cell is activated at the latest stage of the treadmill. If you if you find those cells and sort them out based on their space and where they fire, you can find that when the mouse run through a single lab, you can find cells that are selectively fired at certain locations. It's called place cells. We all know about that, right? But this has been known in hippocampus. That's fine. The question is, if hippocampus is really sending an index to the neocortex, we should be able to see something similar in the neocortex, some information about space. So Dun Mao, a PhD student in the lab, inspired by Jawad work, tried to record special tuned cells, or find special tuned cells in the neocortex. And what he did, he recorded superficial and deep cortical layers using two-photon imaging, and surprisingly, and this is something that was really unknown until that point, that you can find specially tuned cells within the neocortex. Okay. Unfortunately, we were late, so we end up to be the second group to show something in this. So we end up going to PNS. First group went to science. Anyhow, so the idea is that the cells that are basically firing very similar to what we had in hippocampus within the retrospinal cortex. You can see clear index within the neocortex, and this is what it looked like if you sort out the place of the firing of the cells relative to space. Dun also interestingly found that all those cells that are firing at different locations get kind of lose their activity if you lesion hippocampus. And this is a causation that hippocampus sending those index to the neocortex. Okay? Another way is, you could also show that there's a decoding error increase. Decoding error is a fancy method that uses Bayesian techniques to estimate animal position based on their firing rate. That there's a clear increase in error, and that's again when you damage the hippocampus. Ingrid, another postdoc in the lab, shared with Bruce McNaughton, replicated this, but now instead of looking only retrospinal cortex, this area is almost search a lot of places within the neocortex. Okay, so the idea is to bring the mouse every day, but instead of imaging retrospinal cortex, image motor area, sensor area, and many other areas. And do the same thing. So this is place cells, fired at different locations, and this is what it looks like. This distribution of place-like cells in the neocortex, or what I call index within superficial cortical neurons. And when she lesioned the hippocampus, most of those information are gone. except some region still showing some information in the space. And that's exactly was another surprise, because what she found was that there are some distributions, some of the cells that are actually sensitive to cues. It's not surprising for sensory motor area, you know, there's a cues, they sense those information. So they fire, those cells are firing a little bit before the animal reaches the cues, as you can see here. If you lesion hippocampus, those prediction goes away. So those cells fire exactly at the time of cue. So now we at least discovered two types of cells that are basically coding space based on information that is coming from the hippocampus, and cells that coding space based on the information coming from sensory cues. So we call them cue cells. Okay. How much time I have? 10 minutes. Okay. So I'm running late. Aubrey is another PhD in the lab. So what she is trying to understand is trying to find index versus attribute. Attribute is the content of the neuron, okay? The context. So if the animal is running here, so if you basically put two different landmarks or two different texture, so so the idea is whether the animal remember what kind of texture is within the belt. If you replace them, you should be able to, based on neural code, you should be able to differentiate those things. We hypothesizing those differentiations happening within layer 5 neurons. In order to do that, someone has to basically image the whole cortical dynamics within the whole columns, and that's exactly what Aubrey is doing. So imaging all of those at the same time, trying to understand whether she can find index versus attribute within the neural cortex. Anyway, I think that's too much to talk about, and I have very little time, and I don't know whether I should talk about this part or not. So this is data that was generated by a PhD in the lab. Instead of looking at index code at the time of encoding, we looked at the index code at the time of retrieval, when the memory gets retrieved spontaneously within the neocortex. And I think to make the story short, our PhD in the lab recorded the neural activity during the rest. Let the animal run, so you could see all these place cells, and then he clamped the belt, let the animal stay, so he could see same neural activity in rest or post-task period. And what he discovered was that when he looked at the simple hierarchical techniques, you can find ensembles of cells that are getting activated or reactivated together. So there are cells that are activated here, some cells that are activated here, and some cells that are activated here. And when he kind of looked those cells, look at the activity of those cells when the animal was running through the maze, he discovered that by just looking at those activities during rest period after task, you can recreate the entire task. You can basically the same neurons can recapitulate the animals running in the task. So it's kind of reactivation, simple memory reactivation. So forget about this thing, is kind of a statistical thing to show that this is not happening randomly by noise. But you know, again, the things that make his work very different than any other group, or at least what we know about that. So you find two subclasses of ensembles. Some of the ensembles are very narrow field, activation field within certain location, and they are very sensitive to the cues. So we call these cue ensembles. And some cells that are not really having very narrow, they are basically have a bit wider field, and they can basically make a trajectory over the kind of field. So we call this trajectory ensemble. Hypotheses argue that if we have pattern completion, so a small cue, so partial cue should activate index within the neocortex, and the activation of that should basically bring that entire memory back to the animal. And this is what he did. So here he plotted the strength of activation of trajectory versus cue ensemble cells relative to sharper ripples. And what he found was that the cue ensembles were activated before trajectory cells, which is again representing those concepts that We have partial cues that is happening from sensory motor cortex, which is going to the hippocampus sending an index, which is activated in this trajectory, which is able to recreate the entire track in the mouse brain. Anyhow, so this is with too much. This is summary of his finding, and I hope I'm a bit kind of rushing to show a little bit of things that I didn't have planned to show, but I know some of the trainees are interested to a bit of methodology, so I wanted to show some of the methodology that we have developed over the course of past 7 years. So we do wild field imaging, we head fix the animal and record, but that's bring a lot of limitation. You cannot really look at the animal when they are engaged in natural behavior. So we published this paper in Nature Method, in which we basically use some of the kind of camera that you find in your cell phone with a bunch of LEDs and kind of 3D printing. We came with this little microscope that you can put in the mouse brain and now let the animal engage in natural behavior, such as string pulling behavior, which is unfortunately not coming along, to look at brain activity. Now, what we are adding into the circuit is performing imaging also at the same time as electrophysiological recording. So we have done this in past for head fix animal, but now we are trying to add a lot of electrodes while we are imaging the neocortex. So we have units, but also population recording. At the same time, we are also developing this a new small gadget in which allow us to optogenetically activate pattern of region within the neocortex. So we can basically activate or deactivate certain region of the brain while we are recording their activity, which bring a bit of causation into some of the work that we are doing. So I'll just pass by. This is something that I like to show. I enjoyed very much myself when I saw it for the very first time. So this is a mouse that is basically doing, or it's been introduced to string for the very first time in his entire lifespan. So as you can see, just kind of nicely, doing reaching, grasping, pulling out. And this is something that we have been using for the past several years, and we believe and argue that is much better than any other motor learning task that has been out there. And this is a package. If you want to do automatic analysis, it's already available, so you can use it for your recording. And this is Davis. He has a unfortunately, he has a family member with MS. This is very popular in Alberta. And so he was really interested to use this to see whether he can make a bit of contribution to the field of MS. So what he did, he immunized the mouse, they call EAE model, and you forget about the details. What happens when you immunize the animal, after certain days, you start seeing paralyzation of different different part of the mouse, and you start with tail and hind leg and foreleg. And this is a kind of classical way of looking at behavior phenotype in EAE mouse model. So you can see that the animal is showing 12 days after immunization without problems. Okay, what he was able to show that with this technique, this string pulling task, you can basically start seeing the problem at day 4. You can basically really provide this big window of opportunity to see what's really happening with the neocortex and all this opportunity for drug intervention. So this is day 4, and you can see with a bit of simple analysis, you can see that the hands are not anymore coordinately doing that task in this. And what now he's doing is basically using this little mini scope to be able to image the brain activity. What's happening within the neocortex while all these behavior symptoms happening. And again, I argue this is very similar to human doing the same task. So it's little guys, little animals, but they are able to do something that is kind of similar to what human can perform. What I'm though very kind of proud of was to, with the help of student and colleague, develop this what I call high-throughput video-based Homecage system for testing behavior. So we use some of the little tools, and based on requests from industrial funding, federal funding, but they have asked us to commercialize this to be able to sustain it, but it's very cheap, I promise. It's not for profit, it's just to be able to sustain the work. So we came with this concept that we can, instead of bringing the animal outside to study what they are doing, let's just bring the lab inside the home cage. So this is what we have been doing. So now there's a video analysis of the animals, many animals. You can look at their trajectory, you can look at what they are doing, you can look at their feeding, their behavior, you can classify that. You can, the details published in this paper. If you're interested, you can look at the circadian sleep, but not only a small animal, but many animals that can go through different things and different mouse model and so on and on. What we have been doing was to convince a lot of engineering students coming from different universities to Lethbridge. We don't have engineering school there. And the idea is to basically introduce and make gadget that we can basically bring inside the home cage. So this is, you remember that mouse doing a string pulling? So now the mouse is doing a string pulling all the time in the cage to be able to receive a modern world. This is another thing that the animal basically receives some kind of tactile stimulation as a relief for stress. Or now we are also kind of introducing, a tablet based to kind of bring a bit of cognitive task into the home cage. Video analysis is complicated because you cannot sometimes identify the animal ID, so we don't know what animal is doing what, and we can say what is happening. So for that, we really spend so many hours, and this is a big project to come with this UHF-based passive RFID system, in which would allow us to record from thousands of animals, and that would provide us a great temporal resolution. We don't really need, so this would be a great combination of video analysis and RFID system to triangulate the animal position, and that would really increase the scale of the work that we're doing to test. At the same time, in collaboration with these individuals in Laval, we are also trying to record brain activity, but also perform optogenetic stimulation in the home cage. This is a wireless system. So if we combine this behavior phenotyping with this neural activity phenotyping, I think we'll be able to argue that we can basically kind of try to understand how gene and environment could change phenotype or the other way around. So with that, I think I would like to thank again my trainees and my collaborators and the funding for giving us the opportunity to do a bit of interesting things. Thank you.